Okay, I'm going to switch from proteins or particles and adjuvants to vectors, and from B cells to T cells. So what I'm going to tell you today is about a platform technology that we've developed at Okairos, which is based on adenovirus vectors derived from chimpanzees and their use to induce T-cell response in humans. So before doing that, just a short introduction on Okairos. Okairos is a small company which is developing vaccines for infectious disease and, more recently, cancer. We are a clinical stage company. We completed trials in 2 indications, HCV and malaria. And we are located, our labs are located in Italy, even though our headquarters are in Switzerland. It's kind of a strange situation. We are about 21 people, so a small company, and we have received funding from private investors, as well as a lot of support from public organizations. So, why adenovirus and why chimpanzee-derived adenovirus? Well, I think a lot of you will agree that ideal vaccines will have to rely on both cellular as well as humoral arms of the adaptive immune response. And for sure, there is a long-standing experience in vaccinology whereby B-cell response antibodies are very well induced by classical vaccines, protein-based vaccines. But these are really much poorer at inducing T-cell response, and most importantly, CD8 T-cell response. And I think that still today, even with the advent of adjuvants, the best way that has been shown in preclinical models and in humans to induce a CD8 T-cell response is by using the gene that encodes the antigen, which leads to the concept of gene-based or genetic vaccines. And among a plethora of different ways to deliver genes into mammalian organisms and humans, I believe that at least the most consistent data showing good T-cell response and CD8 T-cell response come from the studies that have used adeno5, which has been shown to be, I think, superior to any other viral vector or naked DNA, probably with the exception of DNA endollitoration, which we heard about yesterday. However, you probably know that there are problems with the use of human adenovirus. For example, for human adeno5, which has been the most extensively used in humans by NIH, by Merck, where I'm coming from, there is a problem of pre-existing neutralizing antibodies because of the widespread infection of adeno5 in the human population. And this has shown to dampen the efficiency of inducing immune response in humans. The problem of the other more rare serotypes from which vectors have been derived is that even though they don't suffer from the problem of pre-existing immunity, they are not as potent as adeno5 at inducing T-cell response. And this is shown, for example, in this monkey experiment that was done a few years ago, where we have made vectors, replication-defective vectors from adeno5 and a bunch of rare serotypes, encoding for the HIV gag and injected single injection intramuscular in groups of macaques at 2 different doses, 10 to the 10 and 10 to the 8 viral particles. You can see there is a large difference in the ability to induce T-cell response between the conventional human adeno5 and the rare serotypes. I'll show you more data on this, also on the efficacy of these different vectors. So we thought this is not the right way to go. We should look at a different source of adenovirus, which would provide us for potent vectors that do not suffer from the problem of pre-existing immunity. And to get that, we looked at primates, and more specifically chimpanzees. So we essentially screened biological samples from a large number of chimpanzees from all over the world, zoos, animal facilities, and we identified a large number, isolated a large number of different adenovirus, which we could classify even in different serotypes. And to summarize the work of about 3-4 years, when we were in Merck, we could show in preclinical experiments that these adenoviruses, or vectors derived from these adenovirus, are resistant to neutralization of human antibodies present in the sera of humans. They are capable of growing in already approved cell lines, like, for example, 293 or PER-C6, and they are potent immunogens in mice and non-human primates. This is an example of 2 of the chimpanzee adenovirus vectors, which are compared to the previous set of human. They're at least as potent as human adeno5, if not better. So, another characteristic of these vectors, which we found in macaques, it's very convenient, is the long-lasting T-cell response that they induce. This is an experiment where we had injected one single shot of a chimpanzee adenovirus, a chimpadeno3, we call it chimpadeno3, again encoding GAG, intramuscular, and 6 years later, we could still find some T-cell response by ex vivo ELISpot, 6 years, and we went back into these animals with a different chimpanzee adenovirus encoding for the same GAG antigen to see whether we could rescue in vivo by an amnestic response, a T-cell response, and this is what happened, in fact. So we thought that at that time we had a bunch, a library of chimpanzee adenovirus vectors from different serotypes, very potent, inducing good T-cell response, long-lasting, and capable of being re-expanded in vivo upon encountering of the antigen for the second time. So what we set out when we founded Okairos was to answer to a number of critical questions toward developing vaccines with these vectors, which are listed here. Essentially, do these vectors induce protected immunity, for example, in relevant animal models? Are they safe in humans? Are they immunogenic? And in fact, they are insensitive to pre-existing immunity in humans? Do they induce potent T-cell response, and most importantly, CD8 T-cell response in humans? And also, what would be the most immunogenic prime boost regimen for inducing potent and long-lasting T-cell response in humans? And finally, can we get an early readout on the efficacy in humans? So first question, are they effective at preventing infection and disease in animal models? So I think I'll show you one of the best examples we have gathered in these years, and this is with a candidate vaccine we are trying to develop against Ebola. You know that Ebola infects non-human primates, and the type of disease that is induced in primates is very similar to what is induced in humans, and eventually these animals, which are infected by the real virus, which is taken from humans that have been infected, eventually die if they are challenged with the Ebola virus. So we showed in this relevant model that a single shot of 10 to the 10 viral particles of a chimpadeno3 encoding for the Ebola glycoprotein is fully protective. And I shall mention that this has been done in collaboration with the VRC Center at NIH. And I shall mention that the gold standard adeno5 had already been tested in this model and shown to be fully protective down to a dose of 10 to the 10, same one used here, but not fully protective if a dose would be reduced to 10 to the 9. So we think we have a vector here which doesn't have the shortcomings of adeno5, and at least in this model is at least as potent as adeno5, if not better. And I want to go back to the question of how do other alternative serotypes from humans perform, not only in terms of immunogenicity but efficacy. So here we had a unique opportunity to compare alternative human serotypes with our chimpadeno vectors in this model. This is adeno26, Adeno26 does not protect 10 to the 10. It's 50% protective 10 to the 11, and even at 100-fold higher dose than what we've used with the chimp adeno, it is not fully protective. What about Adeno35? Even worse. 10 to the 11, 10-fold higher than chimp adeno 3, there is no protection. These are data that have been recently published by Nancy Sullivan from the VRC. So I think this is a clear case. We were discussing yesterday about correlate of protection in the influenza section. I think this is a clear case where there is correspondence between the level of T-cell response and efficacy, at least in this model. What about safety in humans? We have conducted a number of phase 1 studies into different applications, HCV and malaria. This is our HCV candidate delivered by chimp adeno 3, same vector I was telling you before. We have tested a number of doses and have observed quite a clean profile, no serious adverse event, and the majority of local and systemic events were mild in nature and only transient. And we got equivalent data with two different chimp adeno, the 3, which has been used for the hepatitis C virus vaccine development, and the 63, which we are using for the malaria program, which I'll tell you about later in the talk, and 4 different antigens in these 2 vectors, and up to a dose of 2 times 10 to the 11, which is higher than what was tested by Merck. And essentially we got the same results, so quite clean. Small studies, but quite encouraging. What about immunogenicity in humans? Again, these are data from our first phase 1 trial for the hepatitis C virus vaccine, the chimp adeno 3, encoding for the non-structural region of HCV. We get single-shot intramuscular values of T cells above 1,000 as an average, about 2,000, in fact, as an average, in 100% of injected individuals, 100%. And most importantly, the frequency and the potency of the T cell induced by the vector is equivalent in individuals that have no pre-existing anti-adeno5 antibodies, as well as in those that have very high titers, 4,000 and above that titer. So that validates the concept in humans that these vectors are not sensitive to pre-existing neutralizing antibodies that are circulating, that are present in humans. What about T cells? These are a couple of subjects in the very same trial. The vector induces very potent CD8, certainly among the best values I've ever seen in humans. What about regimen? I think there are a lot of ideas and regimens being tested, especially for viral vectors or DNA and viral vectors. We did a lot of work in primates with DNA and different viral vectors when we were in Merck, and we continue to do that in Okairos, concentrating on viral vectors. Our first starting point was the notion that you cannot easily re-administer the same vector, particular adeno, several times, because after the first time you already induce high neutralizing titers, and therefore the second administration is not as effective as it should be in boosting the T-cell response. This is shown here in this macaque experiment where we have used the human adeno6 to induce a T-cell response. However, heterologous prime boost with either 2 different serological distinct adenovirus vectors encoding for the same antigen, or for an heterologous prime boost with adeno prime and MVA boost, work very beautifully in macaques. So we thought of bringing both these regimens, the heterologous prime boost in humans, and compare and really have prove in practice whether there is any difference or both could work in humans. And I have to say that up to now, all our predictions based on animal models were very, very, very good in the sense that the potency, the level of immune response, the stability, the quality that we found in mice and in macaques was confirmed in humans by using the same vector. So animal models were very predictive of the human situation up until now. But the situation changed, and we were very surprised to learn that even if we use two serologically distinct adenovirus vectors for a prime boost in humans, you don't get that kind of boosting with the second vector we observed in macaques. And in fact, we don't, even though we get a boosting from the level of pre-injection of the boosting vector, the level of immune response at peak did not overcome the peak response after priming, which is completely different from what we had observed in macaques. In contrast, if you use MVA to boost an adenoprime, the immune response in humans, you do see a higher level of T-cell response after boosting with respect to peak post-prime. Not only that, but if you use the adenoprime MVA boost, what you get at the end is a much more balanced CD8 versus CD4 T-cell response with respect to the adeno-adeno situation. So that's good. So we think this is a better regimen, at least in humans, than the adeno-adeno. I want to share with you some more recent data we got in a recent trial with adeno, chimp adeno MVA for the Hep C vaccine. You know, Hep C is a terrible virus in the sense that it's very variable. It's very comparable, if not worse than HIV. There are 6 different genotypes classified, about 25 to 30 subtypes. Each subtype includes a large number of strains, and even each individual inoculum comes in very many different flavors because there is a hypervariability in some regions of the envelope glycoprotein. So essentially, when you're infected by a swarm of different viruses, and there is a lot of variability around the world. The largest variability is between different genotypes. So obviously, we wanted to test the concept that with one single sequence delivered by this heterologous prime boost, we could achieve a level of T-cell that would be cross-reacting against very diverse sequences of HCV. So we had the opportunity to do this in this trial because we had overlapping peptides covering the complete HCV sequence from very diverse genotypes. This is the reactivity against of our vaccine, T cells induced by our vaccine in humans against the homologous sequence, the 1B, or the heterologous 1A, still same genotype. When you boost and you increase the overall T-cell response, you not only have good cross-reactivity against the 1A, but also, I would say, exceptional reactivity against a very diverse genotype, which is genotype 3, over 1,000, almost 2,000 ELISpot counts. I think this is something quite unique in the field of vector or genetic vaccine vectors in humans. So we're quite confident that this might be a good way to tackle HCV infection in a prophylactic and possibly in therapeutic setting. So we are now aiming at testing this in a phase 2 study in the US, which probably will start second half of this year. Let me spend the last few minutes in trying to provide you with some initial evidence that this approach may be efficacious in humans, not only in animals. And this brings me to another program that we are developing in collaboration with the University of Oxford, the group of Adrian Hill. And this is a malaria vaccine program. We, at variance with others, like for example Glaxo, think that the best way to develop an efficacious vaccine against malaria would be to target different antigens against different stages of the malaria parasite, in particular, not only the sporozoite stage, but also the liver stage. So having this, I think, quite effective way of inducing potent T-cell response, we thought we could use this technology to develop a component for a vaccine against malaria that would target the liver by inducing a potent T-cell response against the liver stage, which would eventually kill infected hepatocytes. So we developed an adeno. In this case, it's a different serotype, chimpanzee-derived. It's called chimp adeno 63, and an MVA vectors encoding for the trap protein, which is a liver stage antigen of Malaria falciparum. And we did phase 1, and again we found that adenoprime MVA boost is very efficient also with these antigens. And here it's interesting, we did also a comparison of what's the efficiency of 1, 2, or even 3 injections of the MVA alone, which shows you that MVA is a very good booster, but it's absolutely not a good primer. That's very interesting. We have recently done the same experiment in phase 1 with HCV and found the same results. So this is not an antigen-dependent phenomenon. MVA is a very good booster. It is not a primer. So we have tested this regimen in a challenge study. As you know, you can challenge volunteers with malaria because there is a very effective drug which can be used in order to prevent disease. And we have shown in this trial, it's a phase 2 challenge study, that the heterologous prime boost, chimp adenoprime MVA boost, is effective at preventing parasitemia in the blood in this human challenge model. And I think this is the most effective regimen with a vectored vaccine against a liver stage antigen that's ever been shown to work in this challenge study. And interestingly, for those people that are interested in correlates of protection, we could find quite a significant correlation between the level, the frequency of CD8, antigen-specific CD8 that make only gamma, not multiple cytokines, only gamma, and protection. So that's a platform technology that we are applying to a number of potential applications. We have a growing pipeline with a few things coming along and are listed here. And I'll stop here by mentioning that a lot of this work was done by collaborators. The group of Adrian Hill for the malaria program, Paul Klennerman, Ellie Barnes, PIs in our HCV trial, both prophylactic and therapeutic. Gary Nebel and Nancy Sullivan are our collaborators on the Ebola program. I didn't talk about the RSV program, which is still preclinical. We're working together with Geraldine Taylor. And influenza, we're working with Susan Epstein and Mary Queeran at FDA. Stop here and take questions if there are any. Thank you.
Any questions for Dr. Nicosia? Please.
Alfredo, nice talk. So, and thanks for bringing up the importance of T-cell immunity. It's very important, of course. So I guess the thing I'm a little bit missing is when you talk about these adenovectors and then MVA boost, so where is the contribution, or what is the contribution of CD4 and CD8? Could it be possible that the efficiency of the MVA boost is primarily due to some CD4T help that then maybe enhances the magnitude of the initially primed CD8 T cells due to their vector? Do you dissect those things? What is the mutual contribution, or?
I can only make hypotheses. We have no data supporting a mechanism. Certainly you do induce CD4 with adeno, except that the response is largely skewed towards CD8 after adeno priming. If you boost with adeno, an adeno prime animal or human, or probably it's the same, you do maintain that different level of CD8 versus CD4, but you increase both. That works in macaques, not in humans. Certainly the MVA looks like more DNA, which has a higher tendency of eliciting a CD4 response. But whether that is, I don't think it's only CD4 that makes the job. It certainly helps.
Question over here.
Again, a good talk, thanks. I'd like to move you a little bit away from the science for a second. You've got a vaccine that has a prime with one vector and a boost with another, and how does the development complexity, the development costs, and ultimately your final cost of goods affect your decisions on what targets you're going to go for in developing vaccines? How does that fit in with the corporate strategy?
It's a very relevant question. You might imagine that we have been asked this by many people. Cost of good. Cost of good at the lab scale, without any optimization for adeno, is very low. We're about 50 cents per dose. MVA looks like the same. It may even improve with the advent of continuous cell lines that are being developed. At the moment, we are making our material in chicken embryo fibroblasts. But there are new cell lines, continuous cell lines that are being developed now. So cost of goods should not be a huge problem. Complexity. Well, virtually any vaccine comes into different vials. And you know, for example, the GSK malaria, the RTSS, it comes into 2 different vials which you have to mix just before injection. And that's GSK. And they're aiming at developing that registration if the phase 3 pivotal trial will be okay. So there is a large experience in administering components, even if they're not mixing into the same vial. So I think that's not hard. Certainly it's more complex than a single injection of one single thing. But you know, I mean, we are talking about difficult targets. We're talking about HCV in the future, HIV, malaria, TB. We ought to be ready to do a little bit more than what has been done in the past.
Final question.
Thomas, Clean Technology. I want to move you back to the science, actually. You said, like, for the malaria model, that it's only interferon gamma, so it's not polyfunctional C cells. What you look like is protective. What's a little bit different, obviously, than some people say it's more important of polyfunctional cells. And the second thing, if you say interferon gamma, did you look at correlates like killing, like, for example, granzyme or real killing assays in that field, if that possibly would be a correlation of protection as well, not just gamma? Okay, so I was talking to Bob Cedar a couple of days ago. He is one of those that really brought up the story of the polyfunction, along with many others. I think the scenario is changing in the sense that the polyfunctionality of T cells correlates with protection, at least in animal models, in some cases in humans, where it has been done, but that's mostly CD4. For CD8, the situation is different. I have to mention, for example, even though that's animal data, macaques, that for Ebola, the situation is very similar. The real correlate of protection is CD8 and make gamma. So it's sort of a recurring theme. We are starting to look at different markers, granzyme, markers of killing. I should say that generally, for what we gather at the moment, there is quite a good correlation between gamma and those markers. So I cannot say whether is gamma or that at the moment.
Let's thank Alfredo again.